Hello and welcome to this Nature Research custom media webcast titled Investigating Membrane Protein Complexes with Mass Photometry. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Refine. Biochemical and structural studies of membrane proteins have long been restricted to their isolated soluble domains. This limitation has prevented the study of complexes involving full-length membrane proteins. This webcast will present recent studies of membrane proteins applying mass photometry to show what mass photometry can do and what makes it suitable for studies of membrane protein complexes. Our speakers today are Dr. Oliver B. Clark of the Department of Physiology and Cellular Biophysics at Columbia University, and Dr. Andrea Saponaro of the Department of Biosciences at the University of Milan. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Oliver.
Okay, so thank you very much, Sarah, and it's great to be here today. I'm Oliver Clark. I'm an assistant professor at Columbia University, and I want to tell you about a study that we've been doing regarding the architecture of the human erythrocyte ankyrin 1 complex, which involves quite a lot of cryo-EM and a little bit of mass photometry during the process. Okay, so to start with, I just want to bring you to sort of the traditional textbook picture of a membrane where we have hydrophobic membrane proteins floating in kind of a sea of lipid. But we know that that's not really what a membrane looks like. If we take a look at this beautiful painting by David Goodsell, where the composition of an erythrocyte is represented accurately in terms of concentration and scale, what you can see is that actually the membrane is an incredibly crowded place. And we have some membrane proteins which are freely diffusing within the membrane and which are unattached to the cytoskeleton and proteins around them. But we also have quite a number of complexes that are restricted in their spatial localization via interactions with the spectrin-actin cytoskeleton, which are actually mediated by a spring-like protein called ankyrin. And we don't really know a lot about the structural basis for that, which is where our work has come in. So what we wanted to take a look at is how are these protein communities or protein neighborhoods organized and tethered to the cytoskeleton, and ultimately, how does this affect the curvature of the membrane? Because of course, in the context of the erythrocyte, erythrocytes have a very characteristic shape, this biconcave disc shape, and that's actually dictated via this interaction between communities of proteins in the membrane and the spectrin-actin cytoskeleton. And disruption of that connection leads to certain genetic diseases where membrane curvature is dysregulated. Okay, so as you'll know, red blood cells are the most abundant cells in the blood and play a critical role in the transport of respiratory gases, taking oxygen from the lungs to the tissues and taking carbon dioxide for excretion to the lungs. And this characteristic biconcave shape provides optimal gas exchange and also allows for flexibility to traverse really small capillaries. And that shape and flexibility is provided by the spectrin-actin cytoskeleton, which in turn is tethered to the bilayer by these spring-like ankyrin proteins. Okay, and if we take a look at what that looks like, so here's a lovely negative stain image from the 1980s, where if you dissolve away all of the lipid in the membrane and just leave the proteins, you get what we call a skeleton. And what you have is this hexagonal organization of spectrin edges here, where at each node you have a complex called the junctional complex that tethers the ends of the spectrin molecules to the membrane and organizes them. And in the middle of each spectrin edge, you have the ankyrin complex, which both tethers the edges to the membrane, somehow affects membrane curvature, and also mediates clustering of functionally related integral membrane proteins, including most prominently the band3 anion exchanger. And I want to emphasize that while we're looking at this in the context of erythrocytes, this is not an erythrocyte-specific mechanism. So in neurons, you also have a spectrin actin cytoskeleton, particularly in the axon initial skeleton, initial segment, and at the nodes of Ranvier, where ankyrin mediates clustering of sodium channels. And similarly, you have the same thing in cardiomyocytes, epithelial cells, and also in skeletal muscle. So we hope that principles derived from our work in erythrocytes might ultimately apply to other systems. And as I mentioned, this ankyrin 1 complex mediates connections between the red blood cell membrane and the spectrin actin cytoskeleton. We know some of the components, as mentioned, band3, which is a 100 kilodalton odd integral membrane protein that mediates exchange of chloride and bicarbonate. Protein 4.2, which is a protein that the function of which is kind of mysterious, but we know that it regulates association of band3 and ankyrin. Ankyrin itself, which is a 200 kilodalton scaffolding protein that mediates the association of these membrane proteins to the cytoskeleton and also mediates their association with one another, their clustering. And then we have glycophorin A, which is a single transmembrane protein that regulates band3 in some sense and may be important for the assembly of this complex. And there are also a number of other potential members, including the rhesus glycoproteins, glycophorin B, and the intracellular cell adhesion molecules LW and CD47. And there's been a lot of beautiful biochemistry over the last 20 or 30 years that's gone into understanding what proteins make up this complex and what happens when this complex is disrupted. And what does happen when this complex is disrupted is that you get dysregulation of membrane curvature. And we know that mutations in a number of components of the erythrocyte ankyrin 1 complex, including spectrin, ankyrin, and band3, are linked to one of the most common inherited anemias, which is known as hereditary spherocytosis. And in HS patients, erythrocytes no longer have that characteristic biconcave disc shape, but are rather spherical. And these spherocytes are fragile and are rapidly degraded by the spleen. So one of the questions here is why are all these very distinctly, membrane proteins with distinct functions clustered together in the same spatial localization? And one of the hypotheses for this that's been proposed is the idea that this complex acts as what's called a carbon dioxide metabolon involved in gas exchange across the red blood cell membrane, where band3 mediates exchange of chloride and bicarbonate, having obvious relation to carbon dioxide exchange, and the rhesus glycoproteins facilitate diffusion of ammonia and have consequent modulation of pH. And we also know that carbonic anhydrase can be associated with this complex, which catalyzes the conversion of carbon dioxide and water to carbonic acid and protons. Okay, so those are things we know, but we don't know all the components. We don't know the stoichiometry. We don't know really how ankyrin binds to the different target proteins within this complex, and we don't know how it affects membrane curvature. So our goal was to elucidate some of these unknown factors by structurally characterizing this complex purified from human erythrocytes. And so that is where mass photometry comes in. We have a very heterogeneous sample. We want to characterize the masses of different fractions during purification. I'm not going to go into all the details of how mass photometry works, but fundamentally, it's a single particle technique that builds on the principles of interference reflection microscopy and interferometric scattering microscopy. And the key thing to understand about it is that mass photometry contrast is proportional to mass. So the scattering signal scales with mass. And that's critical because it allows us to effectively weigh single molecules or single complexes with light with quite high accuracy on the order of 1 or 2%, and over a very large mass range, ranging from around 50 kilodaltons up to the megadalton range. So how do we use that? What does this look like in practice? So this is just a video from Refine showing what the instrument looks like and how you use it. So basically we have a glass slide with little chambers where we apply several microliters of our purified protein sample at very low concentration on the order of a couple of 100 nanomolar. And then we collect data. And you can see that every black dot here represents a single adherence event of a protein complex to the glass slide. And then that data is analyzed, characterizing the contrast of each individual event and putting that together into a histogram, which allows us to see the mass distribution in that particular sample. And the nice thing about this is that that allows us to really analyze a heterogeneous sample and characterize different components within it that have different molecular masses. Okay, so in our case, how does that work? Let me just see if I can advance that. So Francesca Valesi in my lab developed a really beautiful purification protocol for the erythrocyte ankyrin 1 complex, starting from erythrocyte ghost membranes, where first we solubilize them with digitonin, apply them to a density gradient, a glycerol density gradient, in order to separate out the very high molecular weight complexes from the fairly low molecular weight complexes. And then we apply that to a sizing column, a sucrose 6 sizing column. And then we characterize the different gel filtration fractions to identify which correspond to the complex of interest. So if we have a look first at the main peak and have a look at the mass photometry profile of that, we can see that what we have there is approximately a 400 kilodalton complex that looks quite homogeneous, but considerably too small to be the ankyrin complex of interest. It's about 400 kilodaltons, which, taking account for the contribution of digitonin, is too small for anything but just a band 3 dimer. But if we have a look at that little shoulder, we can see that that actually corresponds to a 1.2 megadalton complex. And, doing some back of the envelope calculations, we can see that's more than big enough to be our complex of interest. And although heterogeneous, it does have a Gaussian profile, and it looks like a good candidate for structural characterization. And so in practice in the lab, we use mass photometry in order to screen different detergents in concert with negative stain. So we'll identify different promising looking fractions using mass photometry, and then do negative stain, and then proceed to cryo. Okay, so here's what a representative micrograph from this data set looks like. You can see very nice side views with a micelle here and a lot of ordered mass outside the micelle. And you can see our 2D classes. We have two very distinct populations. We have these larger things, which presumably correspond to that 1.2 megadalton complex. And then we have a few of the smaller population that presumably correspond to that 400 kilodalton entity, the identity of which we'll get to later. And we collected a fairly large data set, about 14,000 micrographs. And from that, we were able to characterize the heterogeneity of both of those species. So the ankyrin 1 complex itself has 6 well-defined 3D classes, of which I'm going to tell you about 2 today. The first we call class 1, which has 3 band 3 dimers, a central RH trimer, and a number of other components as well. So let's just take a look at that in 3D. So here is the ankyrin 1 complex. You can see in the center, so just to orient you, there's the membrane. We'll rotate around. You can see there are 3 band 3 dimers on the outside in mauve, orange, and yellow that surround a core of ankyrin in red and protein 4.2 in green. And ankyrin is making its primary interaction with the membrane via this RH heterotrimer, so this heterotrimeric ammonia channel that's located in the center of the complex, that's comprised of 2 molecules of RHAG and 1 molecule of RHCE. where RHCE, it turns out, forms the primary attachment point for ankyrin. And with each band 3 dimer, we have one molecule of glycophorin A that's attached to each protomer. Okay. So just to take a slightly closer look at the interaction sites for ankyrin, and I should say that all this work that I'll be presenting today, or most of it, is in our preprint, which is online on bioRxiv at the moment. So ankyrin binds directly to the cytosolic amino and carboxyl termini of RHCE via the first 5 ankyrin repeats, which are oriented orthogonal to the membrane, which was quite unexpected. We expected that ankyrin would be a single superhelical entity, and that does not turn out to be the case. Protein 4.2 is oriented vertically with 2 flat surfaces, one of which forms an extensive interface with the initial repeats of ankyrin, while the distal face recruits one of the band 3 dimers. And then we have 2 additional band 3 molecules that interact with different distinct sites on the C-terminal membrane binding repeats of ankyrin. And I should note that none of the transmembrane regions of the band 3 dimers are shown here for clarity. Okay, so one of the things you might have noticed is that the band 3 cytoplasmic domains are unusually mobile. So in particular, the orientation of the band 3.1, we call it cytoplasmic domains that are bound to protein 4.2, are actually inverted with respect to the membrane when compared to the other 2, which is quite surprising. So the 2 that are bound to ankyrin are in a very distinct orientation compared to the one that's bound to protein 4.2. So just briefly looking at ankyrin. So the architecture of ankyrin comprises a common domain arrangement of 24 repeats that form the membrane binding domain, a spectrum binding module and a death domain, and an autoinhibitory motif in the C-terminal tail. And as I mentioned, we see a very distinct reorientation of that first 5 repeats of ankyrin, which orients the canonical protein binding groove to directly face the membrane, we think, in order to bind the membrane-embedded targets, in this case RHCE, and also aquaporin-1, which I'll get to in a minute. Interestingly, if we compare all of the ankyrin binding or all of the ankyrin repeats, if we align all of the ankyrin repeats from ankyrin 1, we can see that ankyrin 5 and ankyrin 6 really are outliers, in the sense that ankyrin repeat 5 is a little shorter than the other ones, and there are also a number of residues which are highly conserved in all the others, which are not conserved in 5 and 6. But interestingly, if we compare 5 and 6 with their equivalents in ankyrin 2 and ankyrin 3, we can see that the linker between 5 and 6 is highly conserved across the 3 ankyrin isoforms, suggesting that this conformational rearrangement might also occur in ankyrin 2 and ankyrin 3, although that remains to be confirmed. Okay, and interestingly, the band 3, we see the N-terminal peptide of band 3.1 coming back and binding in this groove, and it's actually binding in the reverse orientation from what we see for the ankyrin 1 autoinhibition motif, where when that is bound, we see this complete extended conformation. And we believe that that may have some implications for the mechanism of ankyrin 1 complex assembly. I'm going to skip this because I'm running a little bit out of time. Just briefly, the ankyrin 1 complex, as I mentioned, at the core of it is this rhesus polypeptide, or this rhesus channel. So humans have 5 Rh proteins, of which we have RHAG, RHC, and RHD in erythrocytes. And previously, we didn't know the stoichiometry of this assembly. Now we can see that we have 2 molecules of RHAG and 1 copy of RHCE. And RHCE forms the primary attachment point for ankyrin, which are mediated by the N and C termini of the channel. So just very briefly, we have one other class here, which is very distinct, class 2. And in that, we saw this peculiar quatrefoil density, which we were able to identify as aquaporin-1, which was quite unexpected. So let's just briefly take a look at that. So we have here a complex where we again have Rh, we again have ankyrin and protein 4.2, as well as 1 band 3. but we have aquaporin-1 here as well. And if we conduct a local refinement of aquaporin-1 with local C4 symmetry applied, we can actually get very good resolution for the aquaporin-1, allowing us to unambiguously identify it as aquaporin-1 and to assign all the residues in the aquaporin-1 tetramer. Okay, this was not previously known to participate in the erythrocyte ankyrin complex, although there was some fluorescence resonance energy transfer information suggesting that it might be in the proximity. But now we can see that it is part of the complex, and it's located at the mutual interface of band 3, protein 4.2, and ankyrin, the initial repeats of ankyrin. And so this adds another wrinkle to the carbon dioxide metabolon idea, because in addition to the other components I mentioned earlier, now we have aquaporin-1, which we know conducts both carbon dioxide and water, both of which are substrates for carbonic anhydrase. So maybe that has some significance. Just taking a look at the free band 3, that 400 kilodalton complex, which we think comprises free band 3. So that's those red 2D classes that I highlighted earlier. We're actually able to obtain a reconstruction of that initially at 2.8 angstroms. And we can now clearly see the interaction of this single transmembrane protein, glycophorin A, with the dimer. So there's a single transmembrane protein that regulates band 3. It's classically a dimer, but we can see that it binds only as a monomer to band 3. And comparison with the structure of the isolated glycophorin dimer shows that the interhelical angles in the dimer would be incompatible with band 3 binding. Basically, you would have one glycophorin A sitting horizontally in the membrane, which doesn't make much sense. In addition, we see an interesting lipid density of a very characteristic shape that's binding in a positively charged pocket at the intracellular transmembrane dimer interface, which we believe is PIP2. We were able to improve this density from 2.8 to 2.5 angstroms using the neural network picker Topaz, allowing even better characterization of the structure. So just very, very briefly, so we have these two classes. If we align them together, we can see that they're both sterically compatible. So one of the questions is, this is in digitonin. What does this look like in the membrane? So to try to answer that, what we've done is we've extruded native erythrocyte membranes in order to make small vesicles, put those on a grid, blotted away the excess, and then collected tomograms. And with the help of Alex Noble and Jake Johnson at the Simons Center for Electron Microscopy, we're now conducting studies using cryo-ET to try to characterize the architecture of the native complex. And very recently, we've obtained a subtomogram average of the complex at 25 angstroms, and fitting as a rigid body our class 1 structure into the density, we can see that the native erythrocyte ankyrin 1 complex adopts a class 1 like architecture. So that's very exciting. In conclusion, we've solved the structure of an erythrocyte ankyrin 1 complex purified from human erythrocytes, determined that aquaporin-1 is a novel component of the erythrocyte ankyrin 1 complex, and identified RHCE as the ankyrin 1 binding subunit of the rhesus channel, and determined the stoichiometry of this channel. And subtomogram averaging of particles from native RBC membrane vesicles shows that the endogenous complex adopts a similar architecture. So finally, this is the team during collection of this data. It was a really phenomenal project. So we have Francesca Valese here, Zhining Zhang from the Columbia Cryo-EM Center, Laura Yen, who was a rotation student in the lab, and Gladwin Gillis, who was a undergrad student at the time. So finally, it remains to thank everybody who was involved in this work. So from my lab, Francesca Valese has been the driver of the project and really taken this into a remarkable conclusion. Kuk-Ju Kim in my lab has helped out a lot. Laura Yen and Gladwin Gillis also made key contributions. Filippo Mancha's lab has been extremely supportive throughout the project. The Cryo-EM Center at Columbia has been essential in support during data collection and processing. We have a fabulous collaboration with Alex Noble at the New York Structural Biology Center, as well as Tito Kali, who provided the ghost membranes that were used for initial purification. And the consortium on membrane protein production analysis located at NYSBC and headed by Wayne Hendrickson, originally supplied the Refine instrument, which was key in purification of the complex and optimization of purification. And finally, thank you very much for listening, and I would be happy to take any questions.
Thank you very much, Oliver. We will now hear from Dr. Andrea Saponaro. We can see your slides, Andrea. If you could just unmute, and then we will hear what you have to say.
Okay. Can you hear me? Yes.
Yes, we can hear you.
Can you see the screen?
We can see the slides now.
Okay, thanks. So first of all, thanks to the organizer for giving me the opportunity to present my data. I'm Dr. Andrea Saponaro from the University of Milano in Italy. And today I'm going to give you a brief talk concerning mass photometry and how this technique helped significantly our work on ion channels, and in particular in understanding the stoichiometry of association between the pore-forming subunits of ion channels and the regulatory protein. So let me first introduce the channels that we study. These are HCN channels. As you see on the cladogram on the left, they belong to the superfamily of the voltage-gated channels. There are 4 isoforming mammals. They can form tetramers into the plasma membrane, where they conduct a cation non-selective currents. They are expressed in 2 organs in the, let's say, in the nervous system, both the central and peripheral one, where they are contributing to neuronal excitability. And they are also expressed in the heart, where they are controlling heart rhythmicity. That's why they are also called pacemaker channels. So HCN means hyperpolarization activated and cyclic nucleotide regulated channels. As you see from the cartoon, they have a voltage sensor domain that causes the opening in hyperpolarization, as you see from the activation curve on the left. And then they are directly regulated by the second messenger, cyclic MP, which binds to the cytosolic domain of the channel in a region that is called the CNBD, cyclic nucleotide binding domain. And by binding to the CNBD, it causes a right shift in the activation curve, which means that the channel is enhanced during its opening. So cyclic MP, it's an agonist for HCN channel. Specifically for neuronal HCN, there is a brain protein. It's a soluble protein that here is schematized in blue. It's called TRPADE-B. And this protein, as you see from the cartoon, it binds to the cytosolic domain of the channel, and it controls both trafficking and gating. And for what concerns gating, as you see on the left, it antagonizes the cyclic MP effect. So cyclic MP and TRPADE-B, they are mutual antagonists. And TRPADE-B, it's an inhibitory molecule for cyclic MP. We have been extensively studied this interaction between HCN channel and TRPADE-B, but just at the level of isolated soluble domain. In particular, we focused our attention, as you can see, on the interaction between the isolated HCN-CNBD and the region of TRPADE-B that binds to the CNBD and inhibits cyclic MP binding. And to cut a very long story short, we have used the soluble NMR to get, as you see, the NMR model structure of the isolated HCN-CNBD here in gray, bound to the minimal BBB sequence that we call BBB nanopeptide, that by binding to the CMBD inhibits cyclic MP binding. So this is all cool. That we also like to use the knowledge that we get from our structural and functional data to do some synthetic biology. And here, just to summarize the story, we then decided to use our synthetic peptide to control native HCN current in tissues where BBB is not expressed, like in the heart. These cells that you see here is an isolated pacemaker cells of the heart. And basically, we measure native HCN current by applying in the patch pipette our peptide. And basically, these cells, as you can see, they have autocontractions because they are able to auto-generate action potentials. And the firing frequencies of this action potential is under the control of native HCN current, in particular HCN4 current, that is considered the pacemaker current, the pacemaker cell. And basically, the application of our peptide, as you see, significantly decrease the firing frequency because BBB nanopeptide, by inhibiting the cyclic MP effect on HCN current, slows down the firing frequency of these cells. So everything cool, but of course, as you have seen, all these words come from the structural and functional studies of isolated domains of these eukaryotic proteins. And in particular, we have used the soluble NMR and to get the stoichiometry or all the binding features of these two domains, we have used isothermal titration calorimetry. And all these two techniques, they are all they have something in common. That is the fact that you need a large amount of protein. That's why we were working with isolated domains and not with a full-length protein. And this is a limit. But now this is a big limit if you want to then open our studies to the full-length proteins and to understand the structure and the function of the association between HCN channel and its regulator BBB. Thanks to the new technologies, now we can do it because, for instance, we can do structures of full-length proteins by using single particle cryo-EM, and we can also study the stoichiometry of association by using, for instance, mass photometry. What do they have in common, this fancy technique? For sure, they have in common the fact that you need a little amount of protein. Basically, you work with the micrograms of protein, which is 1 order of magnitude less compared to the amount of protein that you need for the techniques that we have seen before, and few microliters, so basically nothing. And for challenging proteins like large eukaryotic membrane proteins and their regulators, these techniques are really opening up lots of opportunities. For instance, for raw concern the structures of the channels, we recently got the cryo-EM structures of HCN4, so the pacemaker channel of the heart. This channel is highly sensitive to cyclic MP. This is the model structure of HCN4 in the cyclic MP bound structure. If I now color the structure based on the root mean square deviation of ITSI alpha atoms compared to the ligand-free state, you clearly see that cyclic MP binding is causing tremendous dramatic conformational changes in the protein, especially at the level of the entire cytosolic machinery. So to cut a very long story short, basically by using single particle cryo, we could finally describe at atomic detail the pathway, the propagation pathway of the cyclic MP from the ligand binding domain up to the end of the cytosolic machinery of the pacemaker channel HCN4, which ends with these two important helices, A and B, A prime and B prime helices, which are crucial for the propagation of the signal from the cytosolic region to the transmembrane region, because as you can see in the quaternary structure on the left, they are located just below the transmembrane region. and they are in contact via two elements, the S4, S5 linker and the HCN domain. They are in contact with the pore domain at the Volta sensor domain. So this is, so basically this just to let you know that cryo-EM allowed us to finally describe the entire propagation pathway of cyclic MP for the pacemaker channel HCN4. So now what about TrpB, the full protein TrpB interacting with HCN channel? As we have seen, many data are just related on the isolated domain of these two proteins, not on the full-length protein. There is just a seminal work of 10 years ago from Zagotta's lab, where they were using single molecule photobleaching to indicate the stoichiometry of association between HCN channels and TrpB. They were using a GFP tag TrpB to try to understand the stoichiometry of association. And basically here on the left, you see the raw data of the steps of photobleaching. They got many photobleaching steps in 4 steps, but also an equal or even larger amount of photobleaching steps in 1 to 3 steps of photobleaching. So even though where they were using different ratio between TRP and HCN2, even in the presence of a large excess of HCN2 compared to TRP. So these data were kind of suggesting that there might be a 4-to-4 stoichiometry of association, but also a large heterogeneity concerning the stoichiometry. So because of this, and since also because we know that in literature, this single molecule photobleaching was used for understanding the stoichiometry of other channels, for instance, for crack channels, and this data based on single molecule photobleaching, they were further disproved by structural data. Then we decided to conclusively get some data on the stoichiometry of the association between HCN channels and TRP protein by using mass photometry. And here, this slide, just to summarize that we have expressed, co-expressed HCN channels, in particular HCN4, with TrpB protein that is fused to 2 tags, histidine tag for purification and GFP tag. These proteins were expressed and purified from egg cells. Here you see on the left the gel filtration of the purified complex, highlighted here in red the peak of the complex. As you see here on the left, there is the Komassi stain STS page and the corresponding Western blot, which basically tells us that the sample is pure, there are no contaminants. There are the 2 proteins of interest, so HCN4 bound to TrpB molecule. Then we use this sample to do mass photometry. We didn't need to concentrate the sample because basically, as I told you, you need very few amount of protein, and basically this is the analysis that we obtain from mass photometry of the complex, and we got a main peak with a molecular mass of 935 kilodalton, and basically we thought that this could be the homotetramer of HCN4 bound to 4 subunits of TrpB. This is because the expected molecular weight, if we have a 4 stoichiometry, is around 800 kilodalton, because the monomeric HCN4 is around 100 kilodalton, and also EGFP TrpB is, as you can see, 100 kilodalton. So we have a discrepancy of 100 kilodalton between the theoretical molecular weight of the complex and the measured one by using mass photometry. But this can be ascribed to the fact that HCN4, being a membrane protein in solution, is kept in solution because it is embedded into a micelle of LMNG CHS. So to confirm that we are in presence of a 4-4 stoichiometry, then we decided to add to this sample saturated concentration of cyclic MP, which, as I said, it's a competitor of TRIP. So cyclic MP should disrupt this complex by creating two peaks. The first peak corresponding to the homotetrameric HCN4 embedded into the detergent micelle, so something that is about 500 kilodalton, and the second peak that should correspond to free GFP triple-B protein corresponding to 100 kilodalton. Indeed, this is what happened. The application of 2 millimolar cyclic MP caused the formation of two distinct peaks, a peak of 100 kilodalton that we assign to free triple-B molecules, and a peak of around 500 kilodalton that should correspond to the homotetrameric HCN4 bound to cyclic MP embedded into the detergent micelle. So based on our data, we could validate finally the previous suggested data that triple-B and HCN4, they have a 4-4 stoichiometry. And the last slide, I just want to give you some hints of why this stoichiometry, what is the significance of this stoichiometry. So basically in neurons, we know that the expression level of HCN at the level of dendrites, it is peculiar, and it follows the expression level of triple-B, because as I say, triple-B controls also the trafficking at the plasma membrane of HCN channels. And of course, this distribution of HCN and of its regulatory protein triple-B can also be reflects also the different biophysical properties of HCN channels along the dendrites. For instance, this is the activation curve of native HCN channels recorded at proximal in proximal dendrites, and this is the curve at distal dendrites. You see there is a hyperpolarizing shift, which is a typical indication of a strong binding of TRIP to HCN channel, which counteracts the endogenous cyclic MP. And why we think the 4-4 stoichiometry is so important? Because HCN channels are important in dendrites to control dendritic integration, which means that they are important to filter all the electrical inputs that from the dendrites they need to reach in phase the soma. So they need to be finally controlled, and they need to be and their response to cyclic MP and to the complexity of cyclic MP dynamics needs to be finally controlled. And TRIP seems to be there for this reason. And in particular, because HCN channels, they have a tremendous cooperativity for cyclic MP binding and activation. Therefore, just a few, 1 or 2 subunits of the tetramers of HCN, if they are bound by cyclic MP, they could cause a full activation. Therefore, we think that because of this, the obligate saturation of triple-B molecules to the 4 protomers composing HCN channel, it is the fastest way possible to control in a strict and fine way the response of native HCN current to the fluctuation of cyclic MP, which are very frequent because cyclic MP, as you know, it's a very important second messenger in all the cells. So with this, I would like to conclude saying thanks to my lab, to the external collaborators, in particular to Oliver and Francesca, because I got this mass photometry data in their lab when I was at Fulbright visiting. And of course, thank you for the funding, and thank you for the attention.
Okay, thank you very much to Andrea and Oliver also. Andrea, if I can just ask you to stop sharing your slides,
Yes.
and then we will have you with us back in the studio. So it is now time for our question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. Okay, so to get us started for today, then I wanted to pose this question to Oliver. Can you explain how you used mass photometry to optimize the purification of the ankyrin complex? You're on mute, Oliver.
2 years of a pandemic, and I still haven't figured out how to use the mute button. Yeah, thanks for the question. So basically, we used it as an aid for detergent screening. So one of the critical things with purifying membrane proteins and in general and multi-protein membrane protein complexes in particular is there's always a balance between finding a detergent that's compatible with purification and finding a detergent that is actually okay for stability of the complex. And in this case in particular, that was really important because with basically every other detergent that we used, we found that elements of the complex fell off. Whereas with digitonin, we were able to isolate a little shoulder on the gel filtration peak, but which we could tell via mass photometry looked relatively monodisperse and had a 1.2 megadalton mass. So that was more or less how we used it, if that makes sense.
Okay, thank you very much. And then the following question is also kind of similar. Maybe you feel you've answered it. Do you think the ankyrin complex is modulated by lipid composition in any way? Does membrane biophysics regulate the complex? Or actually slightly different.
Yeah, no, that's a really good question and something we're interested in looking further at. I can't say too much about that beyond saying that one thing I didn't get into, we identified a PIP2 binding site in band 3 itself, in the isolated band 3. It turns out that's also present in the band 3 that's in the complex. And interestingly, it's located at the interface with protein 4.2 in one class and very close to the interface with aquaporin 1 in the other. So it'll be interesting to see if we deplete PIP2, whether that affects the assembly or stability of the complex. We don't know that yet, but it's certainly an area of interest.
Okay, thank you very much. And now we have a question for Andrea. For your mass photometry, can you calculate binding constants for TRP8B and the channel? What surfactant did you use?
So for mass photometry, we didn't do it. I know there might. There might be a way to do it. They have been implemented some protocol for measuring the affinity. I've seen something. I think in the website of Reframe, you can find some literature concerning the, or even protocol or video also showing how to measure the affinity, the KD by mass photometry. To be honest, I don't know exactly how to do it, but I know there is a way to do it, and it's in the website.
Okay.
We didn't do it by mass photometry. Yeah.
Thank you very much. A quick reminder to our audience, you do still have time to ask a question. To do so,
I have some questions from the audience again.
One moment, I'll come to those.
Okay.
To do so, please just type your question in and we will come to them. Okay, so another question for you, Andrea. Did the HCN channel also have a PIP2 binding site?
Yes, they have. It is not known where it is exactly because there are no structural data available. It is known that they are regulated by PIP2, but there are no data showing where is exactly the binding site. There is no, for instance, a classical positively charged grove, which is typical as Oliver has shown something like this also. A positively charged grove where PIP2 are usually located. In HCN there is no, but we know they are controlled by PIP2. We don't know how. The molecular mechanism is not known.
Okay, thank you very much. I have a further question on the resolution limits of mass photometry, which is a general question, but perhaps we can hear from you, Oliver. What are the resolution limits of mass photometry?
Right. So my understanding is that the theoretical resolution limits are not on the order of 1 or 2% of the molecular mass that you're measuring. But really, I think in many cases, it's going to be limited by the heterogeneity of your sample more than by the resolution of the technique itself.
Okay, thank you very much for that. A further question for Andrea on the problems you can encounter if you record mass photometry data from membrane proteins embedded in detergent. Your thoughts, Andrea?
Yes. Yes, this can be a limiting factor because detergents are quite dynamics and not order structure. So, and this is, let's say, something that introduce in your measurement some uncertainty, because you can be certain about the molecular weight of your proteins, but you cannot be certain about the molecular weight, the exact molecular weight of something that is highly dynamic. What you can do is that you can subtract, you can do a buffer-only measurement with your detergent micelle, and you can see more or less roughly the molecular weight of the micelles that are created by your detergents that you have in solution. And in this way, you can more or less understand what's the size of the micelles that you have. And this is why I was saying during my presentation, the discrepancy of 100 kilodalton that I have between the theoretically calculated molecular weight and the measured one by using mass photometry, sorry, it is most likely due to the micelles in which the membrane protein is embedded.
Okay, thank you very much. We do have a further question that has come in. Does mass photometry data for neuronal membrane protein filming A1 and its interaction with HCN1 conclusively give the idea of membrane potential changes owing to binding and release of the protein complex in vitro? Would this be in accordance with in vivo readings of membrane potential? Andrea,
Yes, can you
can I repeat the
please the
question for
question, because
you?
I did not understand well what's the point.
So the question is asking, does mass photometry data for neuronal membrane protein filming A1 and its interaction with HCN1 give the idea of membrane changes, potential changes that are owing to binding and release of the protein complex in vitro? Are they in accordance with in vivo readings of membrane potential? It's not a very clear question.
No, I don't understand if they are asking if we can do mass photometry on samples isolated from membranes, from neuronal membranes or something like this.
I think so. I think you
I
can go
think
ahead
so.
and
Okay.
answer that
The
question.
problem, I mean, the mass photometry, as we have seen, you can use basically nothing, as in cryo-EM, micrograms and 1 microliter of your sample. Of course, one thing is to purify proteins, even challenging proteins like the eukaryotic membrane proteins from cell line, like egg cells that you can grow in liquid. Another thing is to purify this from neurons. There, the limitation is anyhow the biomass, because anyhow, even to get some micrograms of protein, you need anyhow to grow hundreds of mLs of egg cells, for instance. So I think getting protein directly from neurons, you can do it. I mean, it is possible. It's even more challenging than to get from, let's say, in vitro, as I do. Probably, I mean, we will do it, but we can do it. But yeah, there the problem is that the main limiting step is to get biomass from which you can get the protein.
Okay, thank you very much. And that is where we are going to finish for today. I'd like to thank Dr. Andrea Saponaro and Dr. Oliver B. Clark for being with us today and for answering your questions. I would also like to thank the webcast sponsor Refine and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.
Thank you.